Welcome to the inaugural episode of In Vivo, a channel dedicated to taking all the scientific theory that we learn in class, scientific buzzwords like the second law of thermodynamics and quantum tunneling, to more underappreciated concepts like KEQ and spontaneous lipid bilayer formation, and showing you its place in the real world, how it affects you, your friends, your family, your dog, everyone. We'll explore how life with a capital L uses these scientific principles to solve a variety of problems, from how do I differentiate between self and non-self cells, a question of great importance for immunologists, to even questions as simple as how do I get energy when I eat this apple? Together, we'll develop the concepts necessary for a comprehensive understanding of biochemistry from the ground up, using real-world examples along the way. However, this won't be like any class you might have had before. We'll be learning to walk while we run, meaning we'll explore the basics, amino acids, acid-base chemistry, metabolic pathways, by delving deep into complex analyses of various proteins, protocols, and pathologies. Don't worry though, these videos assume that you have no clue what biochemistry even is. All I ask for is three things: an open and curious mind, steadfast perseverance in the face of challenging new concepts, and a willingness to help others, because after all, we'll be a community who's learning together, and I'd love to see everyone helping each other out with questions in the comments below. This video specifically will be the first in a series titled Protein Profiles. In this series, I'll take an in-depth look at a protein of interest. With that being said, let's dive right in. Ants, worms, spiders, and cockroaches. These insects don't bring up the fondest of mental images or memories. However, the firefly has ingrained itself into the popular consciousness as a symbol of magic, childhood innocence, the arrival of summer, and just plain wonder. You can see that everywhere in our culture, from television shows and movies to music and art. Take a look at this artwork titled Fireflies at Ochano Mizu by Kobayashi Kiyochika. You can definitely feel the sense of awe and wonder through the screen, can't you? These fireflies, or lightning bugs as they're also called, have captivated our minds for generations. But why? The answer, of course, is their lightning. We know nowadays that their distinctive glow isn't some sort of magic, but rather a carefully controlled chemical reaction. Unlike many of the reactions you might have learned about in school, however, this one produces light, or in other words, it's chemiluminescent. Also, since the reaction happens in a living organism, we call the glow bioluminescence, bio for life and luminescence for light from life, if you will. Before describing this episode's protein of interest, I'll delve a little bit deeper into the mechanics of chemiluminescence and how exactly a chemical reaction can have light, of all things, as a product. Now, a detailed explanation of the physical chemistry behind chemiluminescence is outside of the scope of this video, but we can make do with the more rudimentary surface-level view. Now, before I start, I should say that the Bohr model of the atom, what I'm using in my figure, is outdated. Electrons don't orbit the nucleus in predefined paths. Instead, they inhabit regions where they're likely to be. You can thank Heisenberg for this uncertainty. However, the Bohr model serves our purposes pretty well, which is why I'm using it in my figure. Anyways, electrons, shown here in yellow, are elementary particles of negative charge that circle around the nucleus, shown here in blue. The nucleus is composed of positively charged protons and neutrally charged neutrons. Creative naming, I know. The energy level of an electron is directly related to its distance from the nucleus. In this figure, that means that an electron that inhabits the outermost ring, E2, has a higher energy than an electron that inhabits E1. That E on the left-hand side with the arrow is just telling you the same thing, that an electron's distance from the nucleus and its energy level are tightly related. The orbit that is lowest in energy and closest to the nucleus is called the ground state. When an electron isn't in the ground state, it's said to be in an excited state. You may be wondering how exactly an electron can move from the ground state to the excited state. Well, that's where light comes in. An electron can move from an initial state E1 to a more excited state E2 by absorbing a photon, what we call a discrete unit of light, which itself has an energy exactly equal to the difference in energy of the two states, or in other words, E2 minus E1. Chemiluminescence can result from the reverse, that is, an electron can relax from an excited state E2 to a state of relatively lower energy E1 by emitting a photon with an energy exactly equal to, again, E2 minus E1. That photon is what you see with your eyes. That's what you see here on the figure. In a nutshell, that's chemiluminescence. With that out of the way, let's get back to the namesake of Owl City's 2009 smash hit single. As you may have already guessed, I'll be profiling the main enzyme responsible for that glow, that bioluminescence, Luciferase. More specifically, we'll be looking at the wild-type Luciferase from Luciola cruciata, or the Japanese Genji Botaru firefly. That's a mouthful though, so I'll just be calling it Luciferase. Now this is Luciferase. Luciferase is a one-chain monomeric protein. This just means that the protein is composed of just one protein chain, and that its catalytic function, fancy talk for the stuff that it does, doesn't require the presence of any other protein chains. We'll see some more multi-chain polymeric proteins in the future. Now this is a figure that describes this luciferase's specific reaction. It looks complicated at first, but it is pretty easy to break it down. First, we can tell that this reaction's reactants are oxygen, adenosine triphosphate, ATP, divalent magnesium, meaning that it's magnesium ion with a 2+ charge, and something called a luciferin. Let's take a look at the roles of these reactants. We give the name of luciferin to the compound that luciferase modifies and changes over the course of the reaction. In other words, luciferin is the substrate. We call luciferase an enzyme, a protein that does some kind of catalytic function. Wherever you see a luciferase, you'll see a luciferin. The two go together like peanut butter and jelly. ATP is often called the energy currency of the cell, and so it's often used to overcome the activation barrier of the reaction, a kind of minimum energy payment the cell has to give up in order to start the reaction. You'll often see magnesium with ATP, as the divalent magnesium ion stabilizes the ATP molecules, specifically on the negatively charged oxygens. Finally, oxygen gets added to the 5-member ring of the luciferin. After the reaction, you get inorganic phosphate, the PPI, carbon dioxide, adenosine monophosphate, AMP, a proton, H+, oxyluciferin, and of course, light. Inorganic phosphate and AMP come from the ATP cleavage. You can see that after the uppermost step, something was added to the COO- group of the luciferin. That something is more or less AMP, and you can tell that by its characteristic adenine base, marked as AD for space, sugar, and single phosphate group. 3 minus 1 equals 2, and so we're left over with 2 phosphate groups linked together, aka inorganic phosphate. The addition of oxygen in the middle reaction leads to the release of CO2 and H+ via something called decarboxylation, AMP via some sort of cleavage, and an excited form of oxyluciferin, denoted with brackets and a star. It's easy to guess what happens next. The oxyluciferin relaxes from an excited state, denoted by that star, to the ground state. This relaxation results in the emission of a photon. And there you have it, bioluminescence. You might be curious to know the chemistry behind these transformations. This reaction is a perfect example of a redox reaction. Redox, meaning reduction oxidation, refers to the transfer of electrons between substrates in order to form new products. Always keep in mind, oil rig. Oxidation is loss, reduction is gain. In other words, when something is oxidized, it loses electrons, and when it is reduced, it gains electrons. Wherever you have oxidation, you have reduction, and vice versa. The two go together. Luciferase, then, is an oxidoreductase. That is, it's an enzyme that deals with redox reactions. Redox chemistry is an entire field in and of itself, and so we'll cover that more in depth later. Now the fun part, protein analysis. We'll start with the quaternary structure, or rather, the lack thereof. Quaternary structure is the largest level of protein organization and refers to the spatial arrangement of multiple protein subunits that come together to compose a larger, multi-subunit complex. The protein known as proliferating cell nuclear antigen, or pCNA for short, is a great example of quaternary structure. Each subunit here is represented by a different color. Only when these subunits come together as shown do we get the activity of pCNA. It's a key player in DNA replication if you're curious. Quaternary structure also is characterized by a distinct presence of symmetry. You can see here the threefold symmetry of pCNA. Unfortunately, because luciferase is made from only one chain, we don't have a quaternary structure to analyze. Moving on, if quaternary structure dealt with the entire protein complex, then tertiary structure deals solely with one of those subunits, one of those protein chains. In other words, tertiary structure deals with the protein's three-dimensional shape. It won't tell you much about how the protein interacts with other proteins, but it will tell you a great deal about its overall shape and fold in space, and how those two things relate to its function. On the screen now is a surface representation of the luciferase protein, and that bright blue molecule is luciferin. What we can say here is that the surface of this protein is peppered with cavities, and there are quite a number of tunnels that you can see through. Amazingly, it is in these tunnels that the substrate, and only the substrate, as well as a few analogs, but we'll get there later, can fit. This tight fit is due to the various residues and their characteristic functional groups, called side chains, that reside in the tunnel or binding pocket. These side chains interact with the functional groups of the substrate in a way that results in a fit that cannot be matched by any random molecule. This is how the protein knows when the substrate binds, because only compounds resembling the substrate can bind. I say resembling because modern science has moved away from the lock and key model, which stated that for a given lock or protein, there could be only one key, substrate. Nowadays, we adhere to the induced fit model, which puts forward that the lock and key are not so rigid. That is, the tight fit we observe in the binding pocket is a result of complementary interactions between the substrate and enzyme. In other words, the tightness of the fit is directly related to how many interactions the potential substrate can engage in with the binding pocket residues. Any random compound found in the body is not likely to have the array of functional groups necessary for a tight fit. However, the compounds that do have said array will bind tightly and be acted upon by the protein. Scientists, as always, have utilized this theory to create compounds that are an even better fit for the protein than its natural substrate. These often become fully fledged drugs and help treat a variety of protein function-related diseases. So let's start off by looking at the protein as a whole. You can see that there's a lot of protrusions outside into space, as well as a lot of cavities. So let me zoom in right here. And you can see right there's a lot of tunnels that you can actually see through. And it looks like there should be something that should fit there. So here I'm just kind of going around the entire protein and just showing you these different holes right there, these different tunnels. And believe it or not, one molecule is just supposed to fit there. That's your luciferin. So let me go ahead and put it in real quick. And there you go. You can see that it fits right in there. Almost an exact fit. Let's rotate around, see this angle, and you see that it's almost as if these cavities are windows. We'll zoom in a little bit and get a better look at how exactly this fits inside of the protein. Coming around, you can see that these nitrogenous groups, those blue areas, and the oxygens, those red areas, where they bend and crease, those are reflected in the surface of the actual protein. By taking a slice through the protein, we can see that even more clearly. Right where my mouse is, do you see that oxygen? Where it juts into the protein? During some views, as I'm rotating it, you can see that it actually juts into the protein, and that's reflected in the protein surface. The surface has this kind of cave-like shape to it, right there. And those are the interactions between protein and substrate that I've been talking about. Again, we're just looking more into the different sides of the protein, rotating it, and I'm giving you a better and closer view of how exactly it fits inside this protein. We'll zoom out a little bit here so you can get a more panoramic view. And now that should be enough of this surface level view, no pun intended. Let's take a closer look at the actual binding site. And so here you see this electrostatic surface level view. I've taken a slice through the protein, so all you're seeing is the actual luciferin and the surfaces with which it interacts. So as you look at this visualization, just make sure you remember, blue stands for nitrogen, red stands for oxygen, and anything else is represented by white. These amino acids, these molecules that you see in the top right hand corner, those are amino acids that have been characterized by other scientists to have interacted with this luciferin during their experiments. And so you can always tell that this is luciferin by this blue outline that I've given it. And so again, just as I said in the previous visualization, you can see especially right there with those branched tree-like oxygens right there next to that yellow base, that those red oxygens jut into blue areas. We'll see as we turn around. Right here, I'm just giving you a look at the actual tunnel system here, the actual binding pocket, how small it really is in comparison. Keep in mind that these proteins are very small and that these molecules are even smaller. And so having this kind of fit, honestly, is amazing. Just slicing here to give you a better look at this view. And here you can see that sulfur base, that yellow base, and how those two double bonded oxygens jut into nitrogenous territory. Again, a profile view of this molecule and how tight that fit is. And you can see there that the amino acids, those white amino acids, are really close to the luciferin. And one thing to keep in mind is that these active site residues, although close in space in the tertiary structure, might not be close at all in the primary structure, which we'll get to later. Because the protein folds in on itself, these active site residues could actually be hundreds of amino acids apart. Now we're back to the view of luciferase we saw in the beginning of the video. You may have been wondering what exactly those arrows and loops were. Well, they're the secondary structure of the protein, and they come in two main flavors: alpha helices, the loops, and beta sheets. If the tertiary structure was a broad overview of the three-dimensional structure of a protein, then the secondary structure is a closer look at the different patterns we see in that three-dimensional structure. Secondary structure describes how a protein folds in on itself according to hydrogen bonds. These bonds can twist and turn the amino acid chain into loops, or parallel strands, that, when placed side by side, form sheets. The most important characteristic of these secondary structure motifs is that they're hydrophilic. That is, because of the innumerable hydrogen bonds in the center of those structures, they interact very favorably with water. This is why you'll often see alpha helices and beta sheets on the outer surface of a protein, where the water in a cell is, and not that often in the core of a protein, where water generally isn't. So now, here's a view of the secondary structure, but this time you'll recognize that blue molecule as our luciferin. Right off the bat, you can see that the secondary structure motifs, the alpha helices and the beta sheets, they're localized towards the outer edges of the protein. Again, that's where water is, and that's where the most favorable interactions with hydrogen bonds can occur. But as you can see, the ligand binds away from any secondary structure motifs. It binds in the center of the protein. We saw this in the tertiary structure rotation visualization as well. From this, we could make an educated guess. We could say that this luciferin probably has some hydrophobic character, such as aromaticity, or that benzene-like character. If you don't know what benzene is this 6-carbon ring with 3 double bonds, and that's what gives it its hydrophobicity, that lack of O, that lack of any hydrogen bonding potential. Anyways, if we look back at the structure, we can see that we were correct in making that educated guess. All of these rules I'm telling you about follow one another logically, and as a result, we can use them to infer and study different properties of the protein and the substrate, even if they're microscopic and even if we can't see them with our own eyes. We've gone over tertiary and secondary structure, and so now you may be wondering, where's the primary structure? Well, here's the primary structure, or the sequence, of the luciferase protein. Does it look random to you? A random collection of letters taken blindly from a cabinet of amino acids? Honestly, it's anything but. Believe it or not, within this string of letters resides all of the information necessary for the protein to go from this one-dimensional linear chain of peptide-bonded amino acids to a complex three-dimensional structure. I'll show you an example. Now, I've colored each hydrophobic residue or amino acid that prefers not to interact with water red. Our valine, our isoleucines, our alanines, among other amino acids. Do you notice any patterns? For an example, let's look at residues 23 through 38. Seems as though the third of every 4 amino acids along this part of the sequence is hydrophobic. Well, this is a telltale characteristic of an alpha helix. If we check with the sequence and structure catalog, we can see that there is indeed an alpha helix there. Again, because life follows logical rules that play off one another, we can see that although we would think otherwise, the information needed to form a complex 3D structure is in fact right here in this simple string of letters. After all this, you might be wondering, what's the big deal? Who cares about luciferase? Well, luciferase and bioluminescence in general play a great role in nature. First of all, bioluminescence and luciferase by extension produce light for communication purposes. For example, firefly larvae, they produce light to communicate their bad taste to predators. And when these larvae grow up into adults and they have more adult tastes, why they'll use their lights for sex determination to see what kind of sex they're looking at and whether they can mate with them or not. For communication regarding, well, whatever fireflies communicate about, I can't really read their minds. And so one of the last reasons we have bioluminescence, it seems, is that it was a protection against O2 in the early days of Earth, back when it was young, rebellious, and didn't have a lot of O2 around. Back then, oxygen was toxic to a lot of protozoans, fungi, and bacteria. And so bioluminescence, this reaction that uses up oxygen, could have been used as an oxygen mitigation system to decrease the amount of local oxygen and let the organism live. Interestingly, we also use luciferase on the bench in the lab. Right now we're using luciferases and other bioluminescent proteins as molecular reporters. By adding the luciferase protein onto the end of another protein of interest using genetic recombination, we can track its position in the cell by simply adding the substrate, the luciferin, and waiting for the glow. This is called in vivo imaging, no relation. You may recall that the luciferase reaction requires ATP. Scientists have leveraged this requirement to create cell proliferation assays that use luciferase. Essentially, as the population of cells grows, so too does the concentration of ATP. Therefore, we can use luciferase to qualitatively and perhaps even quantitatively examine the growth of cells in culture. Truly, there are a great number of uses for fluorescent molecules like luciferase in research today, and the advances will surely keep coming. And there you have it, luciferase, a bioluminescent protein ages in the making that helps us when we're trying to impress our date by bringing them to a field of fireflies in late summer, or on the bench when we're trying to visualize a protein that we can't see with our naked eye. And that's the end of the inaugural episode of In Vivo's Protein Profiles. Thank you for watching. If you liked the video, please hit like, leave a comment, subscribe, and share it with a friend. If you didn't like it, please leave a comment anyways. I'd love to hear what you have to say, and I'd love to improve in any way that I can. Regardless, please let me know in the comments below what protein you want me to cover next and any other biochemical topics that you would want to hear about and learn about. Thank you, have a great day, and I'll see you next time here at In Vivo.